All right, last biochemistry tutoring of the year. Hold back your tears. Y'all are so sad, I know. Are there any questions from what we've done thus far? I talked to Dr. Sarkar about 30 minutes ago, an hour ago, and he said that the final should be about 25% old material and about 25% from each of the previous exams. So that's what he just told me. I don't know if he told y'all something different. Did he tell y'all anything different than that? But I would rely, I think he usually does less from exam 1 and more from exams 2 and 3. But he just told me 25% from each exam, so straight from his mouth. We've got, my plan for today is to just try to blow through chapters 17 and 18 just to get them done, and then just kind of go back through my old exams. Cool. All right, this needs to turn on. Cool. So like I said, I'm going to try to blow through this as kind of fast as possible. So have y'all gone over chapter 17 yet?
Yeah.
Have y'all finished the book yet? Did y'all go over 18 yet? No? Cool. All right, chapter 17, transcription, synthesis of RNA. What is transcription first off? What does that definition mean? DNA to RNA, yes. So what is translation? RNA into a protein. We need to have those figured out. We need to have those, I guess, sorted through. So this chapter is mainly talking about the creation of RNA. RNA is very similar to DNA. It's pretty much the exact same thing, except it's got a ribose sugar instead of a deoxyribose sugar. Yes, deoxyribose, DNA, ribose, RNA, and it's got uracil instead of thymine. It's got all the other same component parts, just those are the only two differences. Also up here it says that RNA does not necessarily follow Chargaff's rule. Who can tell me why it doesn't have to follow Chargaff's rule? Anybody? Nose goes. All right. So DNA is how many strands? How many strands of, I guess, DNA is 2 strands, right? And they form the double helix. RNA is typically just a single-stranded molecule, yes? Yeah. So the DNA follows Chargaff's rules because both of them have to bind to each other, making that double helix. So RNA doesn't have to bind to anything, so it can just be by itself, so there's no binding rules, so it's just doesn't have to follow Chargaff's. Here's all the same kind of stuff we went over with DNA, just with RNA. RNA is single-stranded. They can loop back on themselves and kind of make a quasi double-stranded molecule. And it has the same thing with adenine forms 2 hydrogen bonds with uracil and guanine with cytosine. So this one's the same thing, AU2 bonds, G, C, 3 bonds. They rhyme, it makes it easy. And RNAs recognize DNAs and RNAs by base pairing. Some RNAs can have a palindromic, I'm going to try this out. Oh, that's awesome. Palindromic structure. What's a palindrome? When we learned that in English class, what is that? Without, maybe you can read it too. Like race car, right? The same thing spelled one way is the exact same thing spelled backwards. Some have these palindromatic sequences, and those produce hairpin-like structures. So one example, like with the rotator, quick, what are the 4 muscles of the rotator cuff?
Supraspinatus, infraspinatus, teres minor, and subscapularis.
Correct. Which one does medial rotation out of all those?
Subscapularis.
Good. All right. That exam's going to be for y'all. Did y'all 90-90 out of that? Can y'all still 90-90 that class or no? It's unfortunate. All right. So if it's a palindrome for RNA, you can read it from the 5' end, 5 to 3' or 3 to 5'. But the main thing is it produces a hairpin-like structure, which looks like that. I don't really use a lot of hairpins, so not really familiar with what that is supposed to be, but hairpin loop. And they also have phosphodiester linkages as the backbone. What types of bonds are formed between adenine and thymine and guanine and cytosine? Hydrogen bonds, right? So hydrogen bonds link the 2 strands together. Phosphodiester linkages are similar to the what types of bonds in proteins? Peptide bonds. Peptide bonds. Some RNAs are biocatalysts. They can catalyze things. We're trying to go a little fast here. If I'm going too fast, just let me know. But I feel like I probably won't be. There's different types of RNAs. You need to know the different types. First one is ribosomal. What do you think it's going to be associated with? Ribosomes, really simple. Associated with proteins, so they help make the ribosomes. We have our messenger RNA, which is what we get when we transcribe from DNA to messenger RNA. It has a poly A tail on the 3' end and a 5' cap containing 7-methylguanosine. That might show up on your test, knowing which side that the poly A is on the 3 and the 5' cap has the 7-methylguanosine. And that's specifically from a DNA. And we have our transfer RNA, which is that cloverleaf structure that we see here on the bottom of the page. We'll get to those in a little bit. We'll watch a little video, so he'll explain it way better than I will. But pretty much what the transfer RNA does is the ribosome reads the messenger RNA, and the transfer RNA pretty much brings the complementary amino acids making our proteins, putting those proteins together. Have y'all had that before in other classes? Or y'all remember Dr. Sarkar saying this? Definitely need to know the difference between those 3, though. So transcription of RNA synthesis. Let's get it on the full page. RNAs are complementary to the DNA sequence. Yes, they need to be. They're formed from them, so it makes sense that they're complementary to them. The only difference, like we've talked about, is that thymine is on DNA, whereas uracil is on RNA. And we can get RNA from either of the 2 strands of DNA, so it really doesn't matter which strand is read. We're going to get both DNA strands are able to create an RNA strand. Transcription of DNA into RNA is catalyzed by the enzyme DNA-dependent RNA polymerase, so mainly just the RNA polymerase. That's probably a test question. Because what was the enzyme that made the first strand of DNA from last chapter? You remember that? Wasn't it just like DNA polymerase? So differentiating between DNA polymerase or RNA polymerase, and there's a couple RNA polymerase 1 and 2 and 3 down here at the bottom. Might just have to sort through those a little bit. Does not need a primer. Did the DNA polymerase need a primer? Yes, it did. DNA polymerase needed a primer. This one does not. And it begins at that DNA promoter region, the same region that DNA polymerase did. This is a little dry. This is like biology early on, I'm sorry. But to make it more fun, I stole Laffy Taffies from Robbie in the cashier's office. So the person who answers the next question correctly is going to get a Laffy Taffy. I'm only 15 weeks late for bringing candy for y'all. I also have grapes if you want a grape. Anyway, so begins at the DNA promoter region. The RNA polymerase attaches to the upstream promoter site toward the 3' the 3' strand and transcribes. I don't know what sigma factor is, but it's something. It recognizes that RNA polymerase is there. RNA polymerase binds to it and starts transcribing. Different ones. Polymerase 1, located in the nucleoli. What does the nucleoli do? Do we remember what the nucleoli does? I feel like it. First chance for candy. If anybody knows? No candy. I think doesn't it help make the ribosomes? Aren't ribosomes made there maybe? I don't really remember myself. I was hoping I'd learn a little bit too, but we'll learn later. In the break, I'll look it up. Synthesizes the precursors for most ribosomal RNAs. The RNA polymerase 2, this one's in the nucleoplasm, which is just the nucleus. Synthesizes the mRNA, right? Our mRNA is made in the nucleus because that's where our DNA is housed, and then it's transported out into the cytosol. And then number 3, also in the nucleus, and makes other types of RNAs and rtRNAs. So transcription, DNA is copied from 3' to 5', but the RNA grows in the 5' to 3'. Which direction does DNA polymerase work? I think it works 3 to 5, but the resulting DNA strand that is created is formed from the 5' to the 3'. Does that ring a bell?
Like synthesis.
So synthesis occurs. So synthesis is always going to occur in the 5 to 3' direction, but it's always going to be read or copied by the enzyme in the opposite direction. So from 3' to 5'. We got it? Okay. And it uses the same as DNA except for uracil. All right. RNA, DNA-dependent RNA polymerases. Adds the ribonucleotide units to the 3' end of the growing RNA chain. So from 5' to 3', it's added to the 3' end. It's initiated by a specific DNA sequence called the promoter. So it starts at the promoter region. And that promoter region just has a TA box or PRIBNOW box. I think that actually might be a test question. What is another name for the promoter region? TA box, or I don't think he'll use the PRIBNOW box. All right. Elongation requires unwinding of DNA. And so our TATA box tells us to start, and then something's going to have to tell us to stop. And where is that? Termination of transcription is signaled by the DNA sequence AAAA, forming a hairpin loop that can lead to transcription with UUU residues. So when we have a UUU hairpin loop, causes the RNA polymerase to release from the DNA, and we're done. We've got our RNA, can go make whatever protein it needs to make. So that's why we make RNAs, because we have all of our genetic information is in our nucleus. Yes, that's where we store it all. But if we need to make proteins, we need to get that genetic information out from the nucleus into the cytosol. And we don't want to expose the DNA to whatever else could be floating around in our cytosol, right? So what happens is we make our RNA to kind of be that carrier of information from the nucleus, the DNA, to making the proteins. That's the entire function of having an RNA. Transcription. Transcription is what again? DNA to RNA. Lappy tappy. Oh, that was a terrible throw. I was an athlete at one point, I promise. So, intron. Here's our coding strand. So our DNA coding strand is the top one. We read from 5' to 3'. Our template strand is 3' to 5'. Yes? So now we need to make our first mRNA. This always confused the freaking crap out of me when I took any of this stuff. And I think I might actually have it figured out eventually after however many years. Our template strand is what we are using to make our RNA from. Okay? That's what we're using. So if we've got a TAC as our first 3, we want to use the opposite of that to make our first RNA, right? Because they need to make those base pairs. T can't just be all by itself, so it needs that A. Then we have an A, then we're going to pair that with a U because it's RNA, and then the C is going to go with G. So it's pretty much that complementary pair. That differs from the coding strand. So the coding strand and the mRNA are pretty much the exact same, minus the differences between thymine and uracil. So the template strand is what we're using, and we're making our RNA off of that template strand. Or you can kind of just cheat and say, hey, all right, I've got my DNA coding strand. I'll just change the T's to U's. But then when we have our protein, and then our protein reads the mRNA, making the specific amino acid sequences with peptide bonds. Questions? Introns, this is a test question, pretty sure. Introns are the non-coding regions of DNA, and exons are the coding regions of the DNA. What does that mean? Exons are what I guess the DNA polymerase actually reads to make an RNA. That's what actually is coded, whereas introns are kind of spliced out. We don't really care about them much. Oh, maybe not. Maybe not. Take that back. Rewind 5 seconds. Sorry. Both introns and exons are transcribed from DNA to RNA. RNA catalyzes the splicing of introns. So both are transcribed, both are made into RNA, but the introns are spliced out. Sorry about that. Got a little bit ahead. So once transcribed, the RNA is processed. Splicing removes the introns and joins the exons together. It's like when your mom makes your, I don't know, like something that you, like one of your foods that you like, but you put in like something that you didn't want to have on there. You kind of just take it out and then you eat it. Maybe that's a terrible example, whatever. I'm okay with it. So the exons are joined together without the nasty asparagus or whatever. Joins continuous sequence, and the 7-methyl guanosine cap is on the 5'. Poly A tail is on the 3'. So, yeah. DNA-dependent RNA is inhibited by actinomycin D. We all know how Dr. Sarkar likes to know what regulates and what doesn't regulate. So actinomycin D intercalates between the DNA strand and does not allow RNA polymerase to move along for a transcription. So actinomycin D does not allow the RNA polymerase to work. Therefore, RNAs are not formed. And if RNAs are not formed, what's going to happen in the body?
RNAs aren't formed, there's no protein.
Yeah. Bingo. Is what's going to happen. We won't make proteins, is what actually happens. But maybe flatulence happens too. All right, what is this picture? I guess here's just an example. Poly A tail is on what side? 3' or 5'? 3', yes. Good. So here's our TA box. Is that, what's another name for that? Pribnal, but more importantly, what's like the other name? What's the region? The promoter region, right? The promoter region. Good. Yeah, I don't know what H and RNA is. I don't like that. Maybe that's human. I'm not sure. Transcription. What I kind of picked out from here was that splicing is accomplished by a special complex structure having catalytic activity called spliceosomes. So they got really original with that one, and that the spliceosome splices. What do they splice out? Introns. And then they join together the exons. And here is a picture of that. And all this happens in the nucleus. And there we go. So it gets rid of all the nasty yellow and puts all the nice greens together. Questions? Sorry, I'm just trying to get through as much as possible. Chapter 18, translation. So protein biosynthesis. So that's when mRNA is decoded and creates polypeptides or proteins. Here's something. The nucleotide sequence present in the mRNA contains code for each amino acid. This is known as your genetic code. I don't think he'll ask that, but he might actually also ask that, so I'm not really sure. One gene may contain more than one codon specific for a protein or a polypeptide subunit. So our genes, one gene does more than one thing. Have y'all heard something different, or have y'all all kind of like, is this all basic knowledge for y'all? So for those of y'all with a biochemistry or a biology compound, that might be something, but this is something I learned. So one gene can do more than one thing. It can make more than one protein. All right, what is a codon? Yes, so a codon is a sequence of 3 bases in mRNA. So we're reading it from the 5' to 3'. We're always reading 5 to 3, bigger or smaller. And it's specific for an amino acid. What happens? Let's watch the video first, and then we'll go through this. Let's do that. Let's switch over. That's not the button I want. That's the button I want.
Translation is the second step in the central dogma that describes how the genetic code is converted. We've talked about how the mRNA codes are recognized by tRNA and how the amino acids are linked together by peptide bonds. A chain of amino acids is also called a polypeptide.
It's like 7 minutes, so.
Polypeptides are assembled inside the ribosomes, which are tiny organelles on the rough ER of a cell. Now that we're learning more about the mechanics of translation, we're going to have to start putting the pieces together. We already understand the role of the ribosome and the amino acids in the process of translation, but how does polypeptide assembly actually occur? There are 3 important steps to the process of translation. There's a beginning step called initiation, a middle step called elongation, and a final step called termination. These 3 words may sound familiar to you. The same terms are used in transcription to describe the steps involved in making the mRNA strand. But here in translation, we're making a polypeptide strand. In either case, we're making a long molecule out of a chain of smaller subunits. So whether we're referring to transcription or translation, the 3 terms accurately describe the mechanics of the process. Let's walk through each step one at a time. We'll start with initiation. During initiation, the mRNA, the tRNA, and the first amino acid all come together within the ribosome. The mRNA strand remains continuous, but the true initiation point is the start codon AUG. Remember that the start codon is the set of 3 nucleotides that begins the coded sequence of a gene. Remember also that the start codon specifies the amino acid methionine. So methionine is the name of the amino acid that is brought into the ribosome first. And how did methionine get itself to the ribosome? By attaching to the tRNA that contains the right anticodon. The anticodon for AUG is UAC. We know that because of the rules of complementary base pairing. The tRNA with the anticodon UAC will automatically match to the codon AUG, bringing the methionine along for the ride. So there you have it. mRNA is attached to tRNA, and tRNA is attached to methionine. That's initiation. The next step makes up the bulk of translation. It's called elongation, and it's the addition of amino acids by the formation of peptide bonds. Elongation is just what it sounds like. A chain of amino acids grows longer and longer as more amino acids are added on. This will eventually create the polypeptide. Now that we've begun with the start codon, the mRNA shifts a little through the ribosome so that the next codon is up for grabs. Let's say the next codon is UAU. So now we need a tRNA that has the matching anticodon AUA. Oh look, here's a tRNA with the right anticodon, and it's brought along a tyrosine. Tyrosine is the amino acid that is specified by the codon UAU. The tRNA attaches to the mRNA in the ribosome and lines up tyrosine right next to the waiting methionine. A peptide bond forms between the 2 amino acids. Then the first tRNA leaves everyone else behind and floats off to find more work to do. Poor methionine, now it's just drifting around like a lonely kite in the wind. That tRNA left methionine hanging by only one anchor, its peptide bond with tyrosine. The tyrosine is still attached to its own tRNA, which in turn is clinging to the mRNA inside the ribosome.
Whoopsie. All right, so that's the beginning of it. Let's get to the rest of it. Any questions on that? We're leaving us a little cliffhanger, but that's okay. We'll answer the question. So a codon, like we said, is the sequence of 3 bases in an mRNA is specific for an amino acid. So like we said, the methionine has a specific codon sequence. Tyrosine has a specific codon sequence. And that's what builds the polypeptides. There are 20 amino acids that we have in the body. Which one is the only one that is achiral? Glycine. We got a Laffy Taffy. Heck yeah. Glycine, right? And are our amino acids L or D? D. Good. But we have 61 codons that represent 20 amino acids, plus 3 stop codons. So what this means is we've got 20 amino acids, 61 different codons. So each amino acid has at least one codon, but some have more than one. I think there's a chart on here at the bottom of the page. We can see here, methionine is the one in the yellow with the start codon here. AUG makes methionine. We can see just below it, though, that GUU and GUC, GUA, GUG all make valine. Excuse me. Methionine is one of the ones that just has the single codon, whereas valine or alanine or asparagine, glycine here at the bottom have multiples. I'm pretty sure he asked us on the exam what AUG coded for. It's just methionine. I think the only ones you'd ever need to know are methionine or the stop ones. So if you ever see AUG, you know that's methionine, that's your start. If you ever see UAA, UAG, or UGA, that's your stop codons. Pretty sure those are the only ones he would ever ask you. Ask him if he ask him in class, because y'all haven't covered this yet, correct? No. Okay. Where else are we at? So we said methionine and tryptophan also only has one specific one. So tryptophan is, where is tryptophan? Tyrosine, threonine, TRY, where is that? If we can find it. UGG. Bingo. Tryptophan. TRP, I'm sorry. And the genetic code is unambiguous, which means a specific codon only represents one single and specific amino acid. So it's both unambiguous and degenerate. Degenerate means, where did degenerate go? Some can be represented by more than one codon, hence the code is degenerate. I don't think he'll ask y'all about that, though. So just, like I said, know the start, know the stop. Genetic codes are not overlapping, because if they overlap, they can't be read correctly. It's like trying to read your cells and tissues book and your DAA book and your biochem book and your DXI book all on top of each other at the same time. It's not going to make any sense. AUG is start. UGA, University of Georgia, is your stop. Mutations. We've talked about mutations before. Yes? We should have. If you don't remember, mutation is when our nucleotide sequence in our DNA is changed, either pretty much by accident or by radiation, what have you. So mutation is just the change in the sequence of DNA, not of RNA. We're back to DNA now, just showing you where we're at. So of DNA, right? So we talked about last time, bless you, when DNA is replicating, right, we can have problems with replication. A mutation is one of those problems with replication, and there's three main different types of mutations. We have a silent, a missense, and a nonsense mutation. A silent mutation is when there is a change in one of the sequences, but we still code for the same codon. This one is starting out UAU, tyrosine, but when it changed to UAC, that's still coded for tyrosine. Going back over here, tyrosine has two different ones. It's UAU and UAC. So even though that changed from a U to a C, it didn't matter. It's still going to make that same tyrosine. Nothing's going to happen. We're never going to know it's there. It's silent. We can have a missense mutation where we change a single base, and that actually changes to a different amino acid. So we started off UAU, and now we've got UUU making phenylalanine. So a missense is where it's like when you're if you are trying to speak a different language, and I guess to give a crude example, instead of saying like año in Spanish, you say ano. So instead of saying year, you say butthole. It's like one of those things. That's what's happening here. Is it coded wrong, and it changed really the meaning of the codon. And lastly, we've got our nonsense mutation, where you accidentally produce a stop codon. And this terminates the strand, and you're done. So silent, you never know what happens. Missense, it's like you misspoke, or you're not making very much sense because you're saying butthole instead of year. Whereas a nonsense mutation, you don't take anybody's nonsense, so you just stop it. Produces a stop codon, and the stop codon is UAG, or one of the other two. Questions on that? There's also insertion or deletion mutations, and the next page is talking about that. And that's where nucleotides are inserted or deleted from the sequence. Bless you. And that can lead to frame shift mutations. Just kind of skipping down to the examples here. We've got this, how are you? Okay. All of our codons are in groups of threes. Yes, from that previous page, it was AUG or UUU or what have you, UGA. So all of our codons are read in groups of three. If we have a deletion mutation where we delete one of the letters, one of the nucleotides, so we delete the W from how are you, that now reads HOA, HOA, RE, O, which doesn't make sense, right? So that's going to code completely different things than what it was originally going to be. Yes? So that's going to be a deletion mutation. Next, we've got an insertion mutation where we're adding this X into between the W and the A. So now we've got how, zar, AU, U. Also doesn't make sense because everything's read by threes. Now it's coding something completely different. So which mutation probably is going to have the least effect on the body? The silent mutation, yes, because it's like nothing ever happened. Which one do you think probably is going to have the biggest effect in the body? Probably the stop, actually, the stop codon, because it's just going to cut that entirely off, and that protein is really not going to be able to do anything at all. If you have a, going back, if you have a missense mutation, you can kind of recover from that. The protein is still going to be overall pretty functional, probably. But but if you have a nonsense, that's the worst one that you can have because it just cuts it off prematurely. Insertion and deletion are pretty bad, but just want to make sure to say the point that the nonsense is the one that's going to have the most global destructive effects. Pairing. So this is what we're talking about in that video. Not we, but the video was talking about between the pairing of the codon and the anticodon. We haven't talked much about the tRNA structure, but we're about to. The codon is on the mRNA. The codon is on the mRNA and is recognized by the anticodon on the tRNA. So the anticodon is located on the tRNA, codon is located on the mRNA. I think that was a test question when I took the exam. So was that nonsense question that I just asked. Which one is the worst one? Need to understand those. Here we've got our tRNA. So where does this come from? The body just has these really pre-made floating around in the body just because if we need to make a protein, right, they're ready to go. They're right there. We have to spend time making them, finding the alanine or whatever that goes with it. Ribosome is reading the mRNA. We'll get to that in a second, but we have our mRNA and our codon. Our tRNA comes on over here with our anticodon, says, hey, you're an AUC, I'm a UAG, let's get together. And so that pairing brings along whatever the amino acid was at this 3' end of the tRNA, and then the next one would come in, make a peptide bond, make our proteins. General structure, just know, I'm pretty sure just know the anticodon, that it's on the tRNA, and that the protein is on the, or that the amino acid is on the 3'. But pay attention to what Dr. Sarkar says tomorrow in lecture. Protein synthesis. I think we only have 1, 2, 3, 4, 5, 6 more pages. Occurs in a few steps. So the activation of the amino acid by, don't worry about that. You initiate the translation, you add more amino acids, making the peptide chain, you stop it, and then you make the protein, all the secondary, tertiary, quaternary structures. Structures, is that the right word? Step 1, activation. Pretty much, you just you read that there's a methionine. Methionine is what starts it, right? Because it's our start codon. This is really simplified, watered down at this point. 2, initiation. And this deals with the sedimentation rates, which you will need to know. The sedimentation, this is probably going to be a test question. Our ribosomes are made up of 2 different functional units. The smaller one, with the sedimentation velocity, I don't know why they describe it by this, whatever. I think it differs from the plant ribosomes. But really what they do is they just put it in a solution and they see how fast it sinks. And the smaller one sinks at 40 S, whatever S is, that velocity, whereas the larger subunit is 60 S. But the place where everyone messes it up is that the combined of the entire ribosome is 80 S. It's only 80 and not 100. That was something that a lot of people missed when I took the exam. So the sedimentation rate of a human ribosome is 80 S and not 100. And that the subunits are 40 plus 60 makes 80 in this case. I feel like you had a picture of a ribosome earlier. Don't know why there's not one right there, but that's okay. Yeah, here's this. Ribosomal RNA. You've got the two types. This is kind of what it looks like, roughly. Together, 80 sedimentation rate. Separately, 60 and 40. What have you. I think the bigger one actually reads the mRNA, and the smaller one deals with the tRNAs, but that also might be backwards. Don't think that's going to be heavily emphasized. All right, where are we at? So initiation, I guess we'll talk about it right now. The 40S subunit, so the smaller one, has 3 sites. I think that actually might be backwards now. The A site for the for where the tRNA binds. Technically, the tRNA is called the amino acyl because it contains the amino group on it. That's why it's called that. The amino acyl tRNA group. Got the P site where the peptide bonds are formed, and the E site, which is the exit site. So A site can also kind of be thought of as the active site. And this is where the video is talking to. So both the A and P site bind those amino acyl tRNAs, and the E site is unchanged. There's a picture. Let's just go to it. Pictures just make more sense to me. Here's our ribosome. And where, oh my God, I can't even read this. So A site, here on the top right, A site is where that first is read. Oh my gosh. Give me a second. I just confused myself in my head. Got too many thoughts running at one time. P site is where peptide bonds are formed. A site binds at this site. All right. The first one is just the weird one. So P site reads AUG. AUG codes for what? It's our start codon, so it codes for methionine. Codes for methionine. So it reads that there's a methionine, so that it says, hey, I need a methionine. So our tRNA comes, brings it, boom, attaches. Anticodon says, boom, here's the methionine. All right. Then our next one, the active site reads, hey, look, all right, I've got a whatever. I've got like a tyrosine, whatever tyrosine is. And then so bring in another tRNA, puts it on the tyrosine. It moves over into the P site, making the peptide bond. Peptide bonds are made. Then after we take off the amino acid, it goes to the E site and then exits, and it's gone. Super simplified, just trying to get through it. Read through that. That will be a decent summary, but y'all need to go over that. Trying to get through it. Termination, stop codons are UAG, UGA, UAA. What else do we have on here? Don't think there's too much on there, but inhibitors, puromycin inhibits peptide bond formation. Chlorem, phenol, and cyclohexamide inhibits the transferase. And the transferase, peptidyl transferase, is when, kind of skipped over it a little bit, but it's when the peptide bonds here are both put onto over here. So this one is when the amino acid is transferred over there. Questions? Go take 10 minutes. Thank you. Okay. Test 1 from 4 months ago. There you go. All right. True or false? Electrons are negatively charged. True. Right? Protons are positive. Neutrons are neutral. True, false. Electrons in the most distant shell have lower energy levels. False, right? They have higher energy levels. Yes? True, false. An atom that loses an electron becomes an anion. Yes, because it loses that electrical charge now becomes more positive. So this is false. Should be a cation. What kind of bond is formed by an attraction between a positive and negative charge? I'm going to start calling on people. Between a positive and a negative charge. This one's going to be ionic, right? This is going to be your ionic bond between like a sodium and like a chloride, making our salt. Our polar covalents are when we make bonds, but they share, or covalent bonds where they share the electrons. But the polar is when they share them unequally, making partial positive and negative charges on our molecules, whereas a nonpolar covalent is where they do share them equally. There's no polarity on them, nonpolar. That'd be like our H2s, our O2s, N2s, such as that. Whereas our hydrogen bonds, right, are from the hydrogen of one atom with an oxygen or a nitrogen of a different atom making those interactions. Water makes lots of hydrogen bonds. That's why it's so awesome and strong. All right, true, false. In a hypotonic solution, the cell will burst. It's true, yes. An acid is a compound that accepts protons or lowers the hydrogen levels of a solution. An acid donates electrons, right? It releases them. It's like, hey, I've got all these hydrogens. You're welcome. And it increases the hydrogen levels in solution. What is the definition of a buffer? Maka. All right, A. What do we think of A? When the pH changes only a little, when a small amount of an acid or base is added by combining with hydrogens. That sounds pretty good, yes? A sounds good. When the pH changes by a large amount, when a small amount of acid or base is added to it. All right, buffers prevent large changes in pH, right? So that would make no sense. That's why A is correct and B is incorrect. The definition is a strong acid and its conjugate base. It's a weak acid and its conjugate base, or a weak base and its conjugate acid. So the correct answers would be A and D. So need to know what the definition of a buffer is. Prevents large pH changes, really by pairing with the hydrogens that are released or gained, resulting in small pH changes and making the conjugate bases or acids. Henderson-Hasselbalch equation is what? It should be C. C. HA over A. Ha over Ah. Should be. So if y'all want to just like, if y'all have your books on you and maybe just like open them up and star them as we're going kind of through these main topics. Because we're about to just work our way through the entire book in an hour, hopefully. or just like the main topics. Which of the following is not an important buffer system? Protein, bicarb, phosphate, lipid. Lipid, absolutely, right? We've got our protein buffer system. Our bicarb is probably like one of the biggest and most important ones in our body. Phosphate, we've got one of those. We do not have a lipid buffer system. That does not exist. Good. What is the name of the functional group? SH. Sulfhydryl. Says it right in the name. Sulfur and a hydrogen, sulfhydryl. Good. What does disulfide look like? Just SS, right? It's just got two sulfurs on it, right? What's an ether look like? Or what was the example that I gave for ether? Ether was like the bat wing one. It's like it make the bat, so it's the it's pretty much just the O, right? Like I said, diethyl ether looks like the bat with the two wings because there's two. But ether is just the O linkage, and then a thioester is what? Is that just so? What? First off, what is in ester? What is an ester? It's like a COOR, right? A carbon. It's like a carboxylic acid, but instead of a hydrogen at the end, instead of COOH, it's COOR, right? So thioester would be COSR, correct? Should we check it? Let's pull that one up. Page are we on? We are on thioester. Hell yeah. Thioester. We've got our COSR. Oh, whoa, that's not on the .cam. That's awkward. It's on page 12 for those of y'all listening. Okay. Chapter 2. What is the pH of human blood? Was there an answer on that one? D. Yeah, that answer choice. 735 to 745, which is slightly acidic or basic? Basic. Our body is basic. True, false. In a chiral molecule, the alpha carbon is bonded to 4 different groups. True, right? It's bonded to 4 different things. Glycine is different because it's not bound to 4 different things. It's got 2 hydrogens on there, making it achiral. And I just answered that question. Which one is not chiral? Glycine. Which of the following is not a branched chain amino acid? Maca. I heard one alanine. That's correct. What's the other one? The other one's proline, right? Because it's valine, has the branch of the V. Leucine, it's kind of like a weird valine. And then isoleucine. Valine, leucine, isoleucine are 3 branched chain amino acids. What's special about proline? Remember, this is the one that had something special about it? Doesn't have a free what? Doesn't have a free what? R group, right? It doesn't have that free R group because it kind of makes that little ring on the side within it. And it's kind of like old back of the brain stuff, trying to bring it to the forefront a little bit. So valine, leucine, isoleucine are our branched chain amino acids. Proline, it does not have the free R group. The tail of the dog, he's chasing his own tail. And then alanine doesn't really have anything special about it. Which of the following is not an aromatic amino acid? D, they all are. Phenylalanine, tyrosine, tryptophan, all amino acids. True, false. Both sulfur and methionine participate in disulfide linkages. False, right? False. Which one is the only one that does? Oh wait, that's a terrible question. That should be cysteine and not sulfur. But that should say both cysteine and methionine participate in disulfide linkages. Come on, y'all. Y'all are catching this stuff? Terrible question. You can call me on it. Cysteine does disulfide, yes or no? Yes, methionine does not, correct? Good. Because methionine does what in a codon that we just talked about? It's our start codon, right? So we don't really want our start codon linking with anything else. I guess it could later, but it's the very end of the tail. Typically, that's not really going to end with stuff, link with stuff. Which contain an amide group. We've got our books out in front of us, so in theory we'll come up with the right answer. Which contains an amide group. Maca. A and D. Yes. What was our rule? That glutamine and asparagine always go together, right? As the same as glutamate and aspartate always go together. So glutamine and asparagine have that amide group, whereas aspartate and glutamate have lost it, and now they just have that COO minus, giving it that suffix ATE. Because that COO minus gives it the suffix ATE. So you know that those two always go together. But so if you see which ones are negatively charged, you're going to go glutamate aspartate, because when you see the suffix ATE, you're thinking negatively charged. Which does not contain a hydroxyl group. Phenylalanine would be correct. All right, so tyrosine has the oxygen in it. Serine, if you drink too much alcohol, you're going to get very calm and serene-like. 3-onine, there's 3 O's in it. One of them's a hydroxyl, leaving out phenylalanine, which is not a positively charged amino acid. Add histidine up there. So which one's not positively charged? Cysteine. Good. What's special about cysteine? It's polar and uncharged. True or false is deuterion. Still haven't figured out how to say that word yet. Can act as only an acid and predominates at a neutral pH. Test taking strategies, if you typically see a word only in true or false, or always in true or false, they're typically usually false. So it acts as both an acid and a base, right? Can exist as both. True or false, at pH 2, glycine will exist in its protonated form. You'll for sure have one of these questions on this test one, right? Something like this, if you're like in an acid solution, will it or will it not have the hydrogen on it? I didn't like those eyes. pH 2, glycine will exist in its protonated form. Honestly, don't remember that answer off the top of my head. I kind of want to say true. But I kind of also, yeah, I want to say that's true. Yeah, it's on the top of page 22. It's got a pH of where it's at. Anything less than 7, it's going to have the proton on it. Anything acidic. So if something is in a acid solution, it will be protonated. If it's in a basic solution, it will be deprotonated. Questions on that? You don't get it?
It's okay. I never got it.
I think it's because it's talking about like the water, like the solution that it's in. I don't know either. I just remember the rule. What kind of bonds join together amino acids? This one should be back of your hand by now. Peptides, right? Chapter 3. Main determinant of 3D structure is, should be tertiary, right? Because primary is just the sequence. Secondary is hydrogen bonds, and tertiary is like the overall structure of one polypeptide strand. A protein's overall 3D structure should be tertiary structure, but we know that quaternary structure is when different polypeptides come together, making that overall structure. Collagen is water soluble. True, false? I think it's true, actually.
Insoluble.
Insoluble?
Yeah, fibrous.
Good for y'all. Collagen, insoluble. Myoglobin, hemoglobin are the ones that are soluble. It's tricking y'all to see if y'all are paying attention. Type of reaction is joins together two different amino acids. What type of reaction? Condensation, right? Condensation makes amino acids at the bottom of page 24. Amino acids are polymerized by condensation to form peptide bonds. So we take out the water, making it rain, condensation. Right-handed alpha helices are less structurally stable than left-handed ones. True, false? They are more stable, right? If you're not right, you're not right. Anti-parallel are more stable than parallel. True, right? Anti-parallel. It's similar to DNA, right? DNA, where they run in anti-directions, more stable. What types of interactions hold proteins' quaternary structures together? MACA. What types of bonds / interactions? So these are non-covalent interactions, so we can get rid of the covalent one. Yes? So ionic, van der Waals, and hydrophobic would probably be the right answer on this one. The main one is that quaternary structures are non-covalent, which will not denature a protein permanently. Which will not denature it permanently. None of the above, right? All those will be permanent. That's why we try to stay away from those conditions. Yes, that's why we don't live on Mars or wherever. Also Pluto, though. Also all the other planets. True or false? Under proper conditions, most unfolded proteins will renature spontaneously. True, right? Innate intelligence, our body's brilliant. It says, hey, I'm broken, but if I go back to the right environment, I can just fix myself. It's incredible. Which is true. Is myoglobin a tetramer? No, it's a monomer, right? Myoglobin monomer. Hemoglobin's major transport is oxygen transport. Major function is oxygen transport. That sounds pretty good to me. Hemoglobin is in the muscle tissue. No, probably because myo, that prefix, meaning muscle, is probably there. So B would be our true answer. True or false? Myoglobin cannot bind oxygen. What page is that on again? 39. Myoglobin cannot bind oxygen, so that is true. Hemoglobin has a higher affinity for oxygen than myoglobin. True or false? True. Actually, it should be myoglobin. Looking at the graph on page 39, we see that hemoglobin starts dumping oxygen earlier than myoglobin does.
Says that myoglobin has a higher affinity.
Myoglobin should have it based on that graph. And where does it say it?
In the chart on page 40.
On page 40, myoglobin has the higher affinity for oxygen. When I write this, my brain started working again. Why is this? Because our hemoglobin takes our oxygen to our cells, so it kind of needs to release oxygen so our cells can keep working. Whereas myoglobin, its major function is oxygen storage. So it's going to hold on to that oxygen tighter. Hemoglobin's taking it and transporting it, so it's more like the delivery man or UPS guy, whereas myoglobin's more of our hoarder, keeping all the stuff for himself. What will cause a right shift in the dissociation curve? I'm sure we loved all these questions. All right, well, do you remember what our rule was? It's going to be an increase in 2,3 BPG, right? So 2,3 BPG, temperature, and PCO2 all go together. So all three of those have to line up. And pH is the only one that's opposite, that decreases. So a decrease in pH is also equivalent to an increase in CO2, temperature, and 2,3 BPG. So that's why D is correct. True or false, fetal hemoglobin has a lower affinity for oxygen than adult hemoglobin. That is false. It has a higher affinity for oxygen. That's on page 43. Has a higher affinity. Chapter 4. Which of the following is not a cofactor? Feel like the answer should be thiamine. Also feel like the answer could also be biotin, potentially. Thiamine would be the best answer here because thiamine is what? It's an amino acid, right? Maybe? Maybe not? Y'all tell me? Yes? True or false, an apo enzyme is made up of a hollow enzyme and a cofactor / enzyme. That is false, right? A hollow enzyme is made up of the apo enzyme and the cofactor. All right, so we need to know the difference between hollow enzyme. So like the hologram is like the functioning one. If we think about the prefix A means without. So it's a enzyme kind of without everything. It needs its cofactor and enzyme. Which of the following is not a cofactor? Again, it's kind of a terrible question. I'm thinking, kind of thinking sulfur. Magnesium's on the chart. Selenium is in the chart. Yeah, sulfur is the answer here. So you're looking for a nonmetal. This class of enzyme catalyzes oxidation reduction reactions. Probably oxido. I spelled that incorrectly. Oxidato. That should be oxido. Reductase, I'm sorry. The conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate is what kind of classification of enzyme? Dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. This one isn't isomerase, right? So what is the enzyme that makes that happen in triose phosphate isomerase? Dihydroxyacetone phosphate, that's step number 5 in glycolysis, yeah? So it's kind of looking back, we can see, if we just remember the enzyme that does that, it kind of tells us the answer, then that's an isomerase. Which of the following catalyzes the formation of various bonds by condensation coupled to cleave ATP? Condensation coupled to cleave ATP. Should also say transferase. All right, so ligases make bonds, yes? Okay, so if we were making bonds, we probably need to cleave some ATP, so this one's going to be your ligase. So that's the only, ligases are the only ones that make bonds. So with this one, the hint here was formation of various bonds as opposed to the breaking of bonds. Hydrolase would be where you're using water to put into it to break it. What is the correct order for the lock and key model? Oh yeah. Oh yeah. What does E and ES, EP, ES, E and P stand for? Enzyme substrate, right? So do substrates come before products? Yes, right? So we want all the P's at the end. Yeah? Do we start out with the enzyme and the substrates already attached, or do they start out separate and then they have to attach? Right? So we have to start out with a plus sign. So it's going to be D. I mean, it's actually really easy. So your enzymes and your substrates are separate, then they combine, and then they make your little product, and then they all separate and go on their happy ways. Km is equal to what? This was actually on our ARE the other day. 1/2 Vmax. Good. 1/2 Vmax. Which of the following is the Michaelis-Menten equation? This one might be a little bit more challenging. But luckily, we have our notes on us currently. The answer is Vmax S over Km plus S. So that's going to be C. So I just remember this one, that the Vmax goes on top, and then there's a plus sign on the bottom. Try to memorize as little as possible for it to stick. What type of inhibition about an enzyme's catalytic function? What type of inhibition affects the catalytic function but not the substrate binding? So our competitive inhibition, they bind to the same spot, right? They're competing for that one spot. So that's going to interfere potentially with substrate binding, right? If the inhibitor's in there, the normal one's not there. Uncompetitive affects catalytic function, but not the substrate binding. So the inhibitor binds to a site different than where the substrate normally binds. So that's going to be the one, the uncompetitive. Whereas non-competitive binds at a separate site, but it can still, but the other one can still, the substrate can still bind to its active site. Maybe go over that, just start that, go over that the day before. True or false, Vmax decreases when Km decreases in uncompetitive inhibition. Vmax decreases, uncompetitive. Vmax decreases, Km also decreases. Good. Last one, which of the following is not an example of reversible covalent modification? So which one's irreversible? What kind of bond can we make that we can't go back from? Should be phosphorylation, right? Because we put on just the phosphate groups. Should be phosphorylation. Yeah. Oh, nope. Examples by reversible. We've got phosphorylation. We've got methylation is one. Adenylation is one. So it should be carboxylation is the one that we're thinking about, right? So when we were in the Krebs cycle and we took off the CO2, we couldn't go back from that, right? Once the CO2, we broke a carbon-carbon bond or we added a carbon-carbon bond. Can't go back from that. So carboxylation is irreversible. The other ones are, you can all go back from.